Hello, everyone. Let's get started. My name is Alan Hudson from the Immuno-Oncology Translational Network Data Management Resource Center. I'm also chair of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. Before we start the seminar, I just have a few bookkeeping items to go over. Questions will be responded to at the end of the presentation. Please enter your questions in the Zoom question and answer box. The audience can prioritize the order of the questions by clicking on the thumbs up button. Closed captioning is available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu at the bottom of your screen. Instructions are also provided in the chat box. If you're having technical issues, you may make a comment in the Zoom chat box. With that, I would like to turn the proceedings over to our moderator, Dr. Brandy Davis-Dusenberry, who is the Chief Science Officer at Seven Bridges, where she oversees all aspects of scientific innovation with a focus on creating solutions that accelerate biomedical discoveries. Brandy leads the development of a multi-collaborative data ecosystems across the NIH and is passionate about working with diverse stakeholders to shape standards for data access, analytical reproducibility, and democratization of cloud access. Of relevance to today's presentation, she's led the Seven Bridges Cancer Genomics Cloud, which is part of the NCI Cancer Research Data Commons since its inception in 2014. Dr. Davis-Dusenberry.
Great, thank you so much. Good afternoon. It's my absolute pleasure today to introduce our speaker, Dr. Andrey Fedorov. Dr. Fedorov is an associate professor in radiology at the Department of Radiology, Brigham and Women's Hospital, Harvard Medical School. Originally from Ukraine, Andrey came to the US to pursue his Ph.D. in computer science at the College of William and Mary in Virginia, which he completed in 2009. His research is in translation and validation of medical image computing technology, clinical research applications with a focus on imaging informatics, data sharing, quantitative imaging, and image-guided interventional procedures. Andrey contributed to a number of open source projects, notably 3D Slicer and Periodomics. He's authored several public data sets aimed to establish best practices in fair sharing of image analysis results. Dr. Fedorov is currently a co-PI of the team tasked with building the NCI Imaging Data Commons, and he'll share an update on this important initiative with us today. Over to you, Andrey.
Thank you very much for this introduction, and thank you for the opportunity to talk about our work that I present on behalf of the entire Imaging Data Commons team. I apologize in advance, I'm recovering from cold, and I may mute myself occasionally to clear my throat. So I will start with disclosures and disclaimers. This work is funded through a contract from Leidos, and the views I present here are views of the IDC team and not necessarily reflect the views of NIH. The team behind Imaging Data Commons includes participants from a variety of academic institutions and also companies that bring together expertise from a range of topics, including cloud engineering, security, imaging standardization, visualization. And the oversight of the project is provided by Frederick National Laboratory and National Cancer Institute. We are also reporting regularly to internal and external advisory boards at NCI, and which include also outside participants. So with that being said, I want to start with kind of summarizing what are those overall general guidelines that we received in order to implement the national cancer data ecosystem. There is clearly a significant interest in sharing data and increasing recognition of the role data plays in developing innovative technology, evaluating disease, and developing prognostic biomarkers. And it was identified that a robust infrastructure to access the data, share the data, analyze the data is urgently needed. So how do we achieve this vision? The way I like to think about this is that we have two very general guiding principles, guiding concepts toward implementing this vision. First is the concept of fair data. The concept of fair data defines, in general terms, what are those characteristics that data should possess in order to be reusable, findable, interoperable, and accessible. And the emphasis here is that we want the data to be possible to be found and processed by the machines. If, for example, you have a data set where in order to understand how to use the data or how to access certain metadata attribute, you need to contact help desk. That's probably not something that machine would be able to do, and perhaps that's not quite fair data. So this publication set up kind of the framework for what it means to be fair. However, how to implement fair principles for specific domain is ongoing work, and that's something that we are addressing in this project. A second guiding principle is that a decision was made to follow the concept of cloud computing, data commons, in order to deliver the infrastructure to the users. So with the traditional approaches to data analysis, you identify the data, you download it to your computer or to your cluster, you install the tools you need to do to analyze the data. And this approach does have advantages because once you configured your system, you can use it consistently, but it creates challenges when you get to a point where you want someone else to reproduce your result, or when you want to share the analysis results you develop locally with others. So with the cloud way, you have virtual, highly scalable, elastic infrastructure that allocates resources as needed for your specific task. It's a place where data can live and be accessed by your collaborators, and allocation where you can virtualize the computational environments so that it becomes a lot easier for others to access it, at least in theory. At least that's the concept. One way or another, these two concepts, fair data and data commons, are kind of the major building blocks that are utilized in the cancer research data commons that is put together by NCI to support cancer research. And probably you've seen this diagram before. The CRDC includes components that store domain-specific data, provide computational capabilities, search interfaces, authorization, and such. And it's very ambitious in it conceptually in terms of trying to address the needs of different sub-communities within cancer imaging environment, those that work with genomics, imaging, and provide integration of this data for those communities across different programs and capabilities. So Imaging Data Commons is this one relatively recent component, data component within broader CRDC infrastructure. And the definition, the way we define ourselves, IDC is a cloud-based repository of publicly available cancer imaging data, co-located with the analysis tools and exploration tools and resources. So I'll try to quickly dig in what this actually means. So by cloud-based repository, we mean that all of the data that we have in IDC is available in cloud buckets. Right now, it's Google storage buckets. We are working to also replicate our data in Amazon. And by doing that, why we are doing this is because there are many tools available in the cloud, both within CRDC and those available from Google that are possible to apply to the data once it's been organized in the cloud. However, at the same time, if you like to use the data locally, you can still download it and use it within your local resources. When we say cancer imaging data, we mean all cancer imaging data. Traditionally, historically, the imaging research has been siloed, and you have community that works on radiology data. They have their own set of tools and conventions. And you have different community that works, for example, with light microscopy data. Our task is to not maintain those silos, but to have a unified repository that harmonizes data under the same data model, using the same interfaces, and provides similar capabilities, general purpose capabilities, to be able to identify and work with that imaging data, cancer imaging data. What are the tools and resources that become available once you have data in the cloud? So there are many big data-oriented tools that are developed and maintained by Google. I will talk about some of those in my slides. We also have tools that allow you to search the data and visualize the data that we maintain. Those tools are usable with IDC data because the data is in the cloud. There is a range of tools and growing set of tools available from CRDC cloud resources. And again, in order to use those, you need to have the data in the cloud. And finally, we definitely want to have tools developed by individual groups, individual researchers, to be interoperable with our data. So all of those are fair game, and we want to ultimately be able to support. So IDC contract was awarded in the summer of 2019. We relatively early followed up with the public release of data. We focused on radiology collections, following our timeline proposed in the contract proposal. And the purpose of the early release was to seek user feedback to guide our development. Production release followed in 2021. And as of today, we have nearly 40 terabytes of data at this point. This data includes radiology and digital pathology, visible light and fluorescence images. And at this point, we have data that comes from different repositories and data coordinating centers. I want to mention that version 10 of IDC is almost ready. We really wanted to have it available today for this presentation, but due to some delays, it will most likely be available next week. So when I talk about H10 today, this is not available right now in the portal that you can access here, but will be available shortly. This is another way to look at the timeline of IDC development. You can see we went from about 1 terabyte of data to nearly 40 terabyte, and the jumps in this timeline correspond essentially to the sizes of collections we ingested. So, for example, here the big jump was contributed by a National Lung Screening Trial collection that alone contains about 10 terabytes of images. And here basically we have the main milestones in our development, various programs and data sets that we included. Here's another look at the facets of data available in IDC. You can see that we cover quite a significant range of cancer types. We have different imaging modalities. Most of the data is from computational tomography, but magnetic resonance and slide microscopy are the next big categories. And availability of data from different modalities, different vendors, different institutions creates tremendous opportunities for evaluating generalizability of AI tools and developing new methods that are robust to variability of data. So what I will do next is go over the main capabilities, what we see as the value proposition of IDC to the end user, what we want you to be able to achieve with IDC. So first and foremost, we want you to be able to find data that you need, the data that's relevant to your research. And we provide a range of tools to do that. You start with our user portal that gives you a quick summary, list of collections, and the exploration portal that allows you to subset data based on the characteristics that we have in our metadata. And in order to facilitate searching, we have a subset of data, subset of metadata fields available in the portal, but a lot more of it is available in the SQL through the SQL interface, which allows you to access all of the DICOM metadata and define very precisely the analysis cohorts. So going back to the guiding principles that we need to follow, the FAIR principle, again, we need the machine to be able to make sense out of the data. And in order to achieve this, metadata must follow consistent data model. So our approach to addressing this for our repository and for our data is to rely on the DICOM standard. And for those of you who do not know DICOM, most likely you experienced DICOM in your life, because anytime you go to a clinic and you get a scan, the equipment producing the scan most likely will generate that data in DICOM format. It's the format that was developed and motivated by radiology clinical applications more than 30 years ago. And over those 30 years, it underwent significant transformations, a lot of adoption, and right now it's used virtually everywhere around the world. However, what is less known is that DICOM is not limited to clinical radiology. It has provisions to handle a variety of types of data. And as we showed in numerous peer-reviewed publications, those capabilities are very relevant for not only for clinical work, but also for imaging research. So we rely on DICOM to harmonize data across different centers, domains, vendors, because very often data we receive is not in DICOM format. And by doing this, we are able to leverage decades of investment into developing tools that are able to talk DICOM. And those include both open source tools and commercially available tools. And also DICOM helps us anticipate unknowns in the future because DICOM is a living standard and is evolving over time to address the gaps that are identified by specific use cases. And by the way, to our knowledge, there is no alternative data model or standard that would allow to bring data from different domains, such as slide microscopy and radiology, preclinical, and basic cancer imaging under the same representation and umbrella. So if we go back to the paper that defined the FAIR guiding principles, and it includes more granular requirements. What does it mean to be fair? In our opinion, with the capabilities available in DICOM, through the harmonization work we put into normalizing the data, and with the features available through the IDC platform, we cover those principles. And of course, there are different ways to look at what it means to be fair. But in our experience, we have not found a better way to achieve this for the imaging data. Here's the workflow of how the data gets into Imaging Data Commons. So typically, the data originally is collected by a data coordination center. And I put this in quotes because IDC does not have a designated data coordination center. Instead, IDC works with established data coordination centers that support individual projects. And Human Tumor Atlas Network, DCC, is a great example. It's up to the data coordination center to establish how to collect the data, how to interact with the sites, how to assign licenses to the data, what metadata to require, and how to de-identify the data. That's a very important and complex topic. Because remember, we are often dealing with data collected for human subjects, and we have to be respectful and very careful treating this data and making sure that public data sets do not compromise patient identity. Once DCC collected the data set, it interacts with the Imaging Data Commons team. And at this point, we review how data was de-identified. We reach agreement that those procedures are satisfactory to the IDC team and to NCI. And if necessary, if appropriate, we work to harmonize data into DICOM representation, if it's not already in DICOM. And you will notice here this blue arrow. And the reason is that based on our experience, very often the case is that the data is not born fair. The data may not have all of the metadata that it should have in order to make sense out of data. And it does happen that the data as produced by DCC may have gaps. In this case, through DCC, we sometimes need to reach back to the original data submitters to refine the data, to add missing components, and to make sure we have all of the metadata to before we can share the data set. And once this step is done, release is very easy. Yeah, so these steps are quite straightforward. We go through the procedures to release both the files that we converted, and we make the metadata available. And metadata is absolutely critical because if you think about 40 terabytes of data, millions and millions of files, you really need an index to navigate and narrow down to the specific files that you need. Because of that, we pay so much attention to curating the metadata and developing interfaces that enable users to interrogate that metadata. But DICOM is a lot more than just a data format. It does normalize image pixel representation and metadata, both for images and image-derived types of data. But it also defines procedures, how to perform de-identification, which attributes have to be adjusted when we want to remove patient health information. It is evolving closely in collaboration with other relevant standards. As I mentioned before, it's a living standard. It's been refined continuously. And very importantly, it provides the definition of the standard API, standard web interface that can be used to access the data. And this is very important because in our work, we don't want to implement every single component of the infrastructure. We want to leverage as much as possible the work and effort put into developing and testing tools available from the industry and from the open source community. So let's say you found some images that some data that is relevant for your work. What we want you to be able to do next is to explore this data as much as possible, make sense, understand this data as much as possible without having to download anything to your computer. So in order to do that, we provide you with the viewers that allow you to look at the images, look at the annotations. We maintain different viewers for radiology and pathology to meet the community-specific requirements. And you can also use the interfaces we provide, for example, to find related data. So you can, if you found a slide microscopy image for a patient that had prostate cancer, you can see, oh, does this patient also has magnetic resonance imaging, for example? And does that magnetic resonance images, do they have annotation of the tumor in that image? So you can find that related data and understand better what is available for you as you develop your analysis tools, or if you want to evaluate some existing tool. You also you can see what is the heterogeneity of the data available in IDC. So for example, if you developed an algorithm that appears to be very successful in segmenting lung nodules for the data that you collected at your institution, you can go to IDC and find lung CTs from a variety of vendors and a variety of groups that collected this data. And you can often find also definitions, annotations of those nodules in IDC. So you can evaluate generalizability of your algorithm on this quite large data set. And by the way, if your algorithm is still successful in your judgment, you can take the annotations produced by your algorithm and contribute them back to IDC so that other users can really benefit from your work. So here's another visualization, which is something that's coming up in IDC release version 10, which is the visualization of a sample image from the Human Tumor Atlas network data release. So you can see that in this image, you have multiple channels, and this is just a subset of channels that are available in the data. And with the viewer that we provide, you can adjust the visualization of individual layers, color, opacity. You can relatively efficiently navigate this image. And the capability we provide here is backed up by the same DICOM server backend from Google that supports radiology data. And the data and the metadata follows the same data model that we use to harmonize images that have very different attributes than H10. Apologize. All right. So how do we achieve this? Behind the scenes, the IDC is powered by Google Cloud Platform. And Google Cloud Platform provides that implementation of highly scalable, elastic, secure, and reliable infrastructure that can provide you with compute, network, storage, but also a lot of very powerful services, such as implementation of the DICOM web interface and interaction with the DICOM metadata extraction. So we benefit a lot from the availability of Google Cloud Healthcare API, which provides the virtualized concept of a DICOM server. So you can initialize a DICOM server with a few clicks of a button. You don't need to worry what should be the performance characteristics of the machine where you run this server. You don't need to worry about what to do when your data needs grow 10 times more than what you have right now. All of this will be taken care of by the cloud. And you can enjoy accessing this data through the standard interface, which is not proprietary, but open DICOM web interface that is also implemented by the open source viewers that we utilize to interact with the server and provide visualization. What we also get from Google Cloud Healthcare API is automatic extraction of DICOM metadata into BigQuery tables that are then available again through the standard interface, in this case, SQL interface, that allows you to build a simple or complex query, as complex as you need to identify relevant information in the IDC data. These are the viewers that we utilize in IDC. For visualization of radiology data, we rely on Open Health Image Viewer, OHIF. It's a very popular project that has been funded a lot by NCI through grant mechanisms. And for the pathology, we rely on the Slim Viewer developed by the group of Marcus Herman at Mass General. And Marcus is also a member of IDC team. I want to emphasize that we don't lock content modifications to the viewers that we do to satisfy IDC needs. Everything we develop in IDC goes back into those open source projects. And in fact, we have instructions how you can take off-the-shelf OHIF viewer, how you can populate your own DICOM store within Google with your own data and connect your instance of the viewer to the data. So all of the components and technology we use in IDC is easily transferable to help you do similar things for the data that is not public. And I think there is a lot of power to that concept, because you don't need to worry that you invest in a lot of effort learning this technology, which will only, and this knowledge would only be applicable in IDC. This knowledge is transferable, and it will help you in many, many other projects if you work with cancer imaging data. The Google BigQuery deserves, I think, additional attention, because the opportunities it opens really to navigate this very complex metadata available in DICOM are truly unprecedented, because the metadata tables that we have for our data has millions and millions of rows and over 500 or 600 of columns. And very often the metadata that is stored in DICOM is highly hierarchical. So extraction and navigation of this data is very difficult if you need to do it yourself without a hosted solution. With BigQuery, you can use a standard query language, SQL, to write those queries, and you can leave it to IDC to do data curation and ingestion. And here is a kind of a use case example to give you some idea why this is actually important. So National Lung Screening Trial is one collection that I mentioned in the past. It's probably one of the largest collections we have in IDC. And it contains data from a variety of hospitals, mostly computed tomography data, but also pathology. So if you want to subset that data, for example, if you're interested in a specific reconstruction kernel type, or if you want to look at specific image resolution without Imaging Data Commons, what would you do? You would have to download 11 terabytes of data. So you would need to find the disk, you will need to spend time, you will need to use the tool to download it. You will need to extract relevant metadata from those 11 terabytes of data. So for that, you will probably also need to write some scripts. You will need to analyze the results of that extraction. You will need to establish mapping between the metadata you extracted and the individual files. Probably you would want to examine the quality of images. You would need to look at the images before finalizing your cohort. So you will need to maintain and install the viewer. So all of those steps you would need to do on your own. If you want to do this kind of subsetting with IDC, you run a relatively simple SQL query. You can view any of the studies, any of the images without writing any code. So you can select what you need, and you can have pretty good confidence that the images that you selected, what they are. Do they have artifacts? Do they have missing slices? All of this you can do without having to download any single file. And this is the query that you would need to write. It's really not that complicated. Hopefully, even those who never saw SQL before can appreciate this. So basically, what we do, we select study instance UID, which is a unique identifier that refers to the DICOM study. And the second attribute is the URL that points to the location of the file in the cloud, in the Google bucket. So we select these two columns from this metadata table that IDC curates. And we say we only want to select this column for the rows where modality is CT and collection is NLST and slice thickness is less than 1. And that's all. And once you run the query, if you are satisfied with what you see in terms of images, you can write the result of this query into a manifest. Once you have the manifest, you can feed the manifest into the download tool, and you can download those files to your computer or to the cloud virtual machine and do the analysis. So this is it. These two steps: create the manifest and download the content of the manifest, and you have the files specifically that you selected. So if I had to say why IDC exists, is that at this point, I strongly believe that IDC is a unique resource that provides capabilities to visualize, search, and subset this highly versatile 40 terabyte data set of public cancer imaging data. And we hope that these features I showed to you already resonate with at least with some of you and you find useful. But this is not all that IDC provides to you. Once you identified items that are of interest to you, you can save this manifest. You can save the list of subjects or images or studies. And we, as IDC, give you a promise that unless some of this data needs to be pulled due to PHI concerns, the data that you see in IDC today will be there as long as we exist. So you can, the reason it's important is because imaging data is not static. Very often there are collections that are revised. There may be a new version of the image that was changed in some way by the contributor or by the data coordination center. With IDC, we maintain versioned copies of the data. So what you see today will also be available 1 year from now, as long as IDC is around. And this is very important because for those of you who develop image analysis tools, reproducibility is a big issue. And more often, reviewers of the manuscripts, grant reviewers, ask questions: how do we establish that the data you used in training does not overlap with some other data set? How can I reproduce the data you report? How can I make sure when I train my algorithm, the data I used to train it does not clash with the other data set you used? And IDC provides answer to these questions. So instead of verbally discussing how you selected the cohort for your training, you can actually have an ambiguous list, fixed list of items defined by unique identifiers that shows this is the data set I used for model training, this is my test cohort, this is my validation cohort. And anyone can, at any time, download those exact files, run your code, and reproduce your findings. We provide API to work with those cohorts. So anything you can do in the portal, and mostly related to creation of cohorts, can also be done via API. And API is not intended to replace the BigQuery API. BigQuery API is the go-to solution for metadata exploration, but instead API is mostly for you to support your cohort operations. So next thing that you can do, you can take the data that we curate, and you can use existing cloud tools to work with that data. And here I list just some of the tools that are available from Google. For example, Data Studio is a very powerful tool to build interactive dashboards. So for example, if you have a need to explore certain metadata attribute that is not available in IDC portal, and at the same time you have a customer that or user within your lab, for example, or API who doesn't have time to write SQL queries, you can, as for example, investigator or student, write those queries, create interactive data portal using Data Studio, and focus on specific aspects of the IDC data and share it with your collaborators. You can deploy desktop tools on the cloud VMs, and we do have documentation examples how to deploy cloud VM with 3D Slicer running, for example. And you can use that recipe to take any desktop-based tool and make it available through the browser interface. So you can follow your conventional approaches to working with data. I have to admit that integration with the CRDC cloud resources is work in progress, but it's definitely a very important topic, and we are continuously working to make sure that CRDC cloud resources can also work with data in IDC. Over time, what we discover is that cloud and on-premises approaches both have advantages and disadvantages. Where we believe cloud-based approach really shines is in enabling reproducible science, because with the virtualization, you can really establish consistent environment for the virtual machine you use in analysis. You can execute precisely the processing steps, and with Imaging Data Commons, you can also freeze the data you are analyzing in your workflow. And over the past years, we've been working on several examples in radiology and digital pathology to understand how to develop those reproducible workflows and how to share them with the community. And the tool that we believe is kind of the easiest to get started with the cloud is Google Colab. So Colab is an environment where Google gives you for free a virtual machine that can have a GPU. And you don't need to commit your credit card. You don't need any registration. All you need is activation of Google Cloud, and you need to have a Google account. Once you activate that virtual machine, you can write those interactive notebooks that combine text and code, and you can execute the cells of this notebook in the cloud. And you can easily share these notebook artifacts with your colleagues. So this becomes very easy to, for example, make your analysis tool available for reviewers of your paper. So once you have some notebook that you think reached some level of maturity, you can save it to GitHub, or you can start from a notebook available already in GitHub and shared by somebody else. Your notebook cells can include installation of prerequisites from, for example, Python packaging repositories, and IDC can provide you with the capabilities to subset the data through SQL, BigQuery Python API, download the input data, and also visualize those samples that you are analyzing. And in the end, this notebook can produce outputs that you can submit, for example, to a data coordination center or to the repository that you control. And what is interesting is that if at some point you run out of the capabilities that are available in the free tier of Google Colab, there's also a possibility to connect Colab to a custom compute engine. So there is a way to go from this toy example to very complex and very powerful setup. And a big kind of a source of reluctance for users using the cloud is the need to commit the billing account and a lack of clear understanding about the costs. With Google Colab, you can control costs very easily because there is a free tier, and then there is a fixed cost per month for different non-free tiers. All right, so if you if you follow our documentation, we have several examples of Colab notebooks that hopefully are easy to follow, and we are very interested to get your feedback. What are the missing pieces, and how to make our content more accessible and clear. Finally, last but not the least, really, is a lot of data in Imaging Data Commons has components, non-imaging components available within other repositories within CRDC. And emerging capabilities within CRDC, specifically Cancer Data Aggregator, allow to query CRDC as a whole and identify, for example, which of the subjects that you are analyzing and that have imaging data also have genomics, proteomics information. So this is very exciting capability that is available right now in CRDC. One of the most frequently asked questions is, how do I get my data into IDC? And I want to say that IDC right now, at least, does not operate as a repository that takes any data from anyone, any individual researcher. The main concern here is to ensure that the data is properly curated and de-identified and has scientific value. This is a kind of a work in progress procedure for data ingestion by IDC. Up to this point, we only ingested data from specific designated data coordination centers. And for every data coordination center, we have to amend our security paperwork and have an agreement with those centers before we can ingest their data. But we are open for discussion, and we are working to refine this process. And definitely our goal is to make as much data as possible available within IDC. So I'm almost done. I know I'm a bit over time, but I want to just talk a little bit more what I think are the main challenges that we are facing right now, and what are the next steps that the way we see them. So I think these two guiding principles that we established in the beginning of this presentation are also the main challenges that we are facing. Yes, the concept of fair data is very nice, and it brings a lot of promise. But the reality is that you almost never get fair data from the submitter. For the submitter to generate fair data is a lot of additional effort, and the benefit for the submitter is not always clear. You need to invest a lot of time, and why this is important is need to be communicated to the submitter. And a lot of this work needs to be done by data coordination centers and by the Imaging Data Commons. And this inevitably introduces delays and increases complexity of data in some way. And also the process of making data more fair arguably never stops, because when you have a user that tries to interact with your data and they have troubles navigating something, or maybe there is some inconsistency that was produced by the image acquisition equipment, this arguably requires remediation. Also, our approach, as I argued, for using DICOM to harmonize the data, yes, we believe that there is no alternative to using DICOM if we want to harmonize data from those different silos and put them under the same data model. But the reality is that DICOM is a complex standard, and it requires effort to understand and learn how to use it. So that's the first challenge about the fair data. Second challenge, I think, is overall the use of the cloud. The reality is that as a community, most of us within institutions have on-premises resources. We have clusters, we have IT infrastructure at institutions that's supported from the indirect costs of the research grants. We already know how to use this infrastructure. We have incentives from institutions to keep using that infrastructure. There could be security concerns from the institution to do research in the cloud. So that's the reality. And another part of it is that it's by far not always clear, is it economically beneficial to actually switch to the cloud if you take all of the other things out of the picture? It's often not trivial to know how much it will cost to complete a specific task. Yes, indeed, you pay only for what you use, but sometimes the reality is it's difficult to know what will you actually be using. It's very complicated. The cost estimator calculators are quite complex. And what cost estimator calculators from Google and Amazon, for that reason, do not take into account is that the users will need to first learn how to do things. And you cannot put a cost on that. The users will mess up. The users will make mistakes. How to deal with this, it's not clear. And cloud resources within CRDC do help with that by providing guardrails and more tightly controlled environments. So we need to find the middle ground between using on premises for some tasks, using the cloud for other tasks, and educate the user how to benefit from the cloud. So I think the priorities, the way I see them, are in the context of the challenges that I believe we are facing. Yes, of course, we will continue working with new data coordination centers. We will continue growing the data offering that we provide in IDC. But at the same time, I think it's very important to not just do ingestion of new images, but also see how can we enrich the data already in IDC. So for example, if you have a CT image and you are genomics bioinformatician, it's probably not actionable to you. But if you have a CT image of a liver tumor where you have the tumor region well defined and you have radiomics features extracted from that image, that becomes actionable. And that becomes something that even genomics bioinformatician that without imaging domain expertise can correlate with genomics findings. We should spend a lot more time on user outreach to address those challenges that I mentioned before. We need to educate users how to use IDC, DICOM, cloud, and we put a lot of emphasis into this. Partnership with CRDC components is key. We have been working with cloud resources and CRDC DCF, and we are still having unresolved issues related to indexing of IDC data that we need to resolve. And finally, I think partnership with the commercial entities is very important, because without this very robust infrastructure developed by commercial entities, it would be difficult to make CRDC a reality. As I mentioned, we are working on replicating IDC data on Amazon Cloud and continue work with the CRDC cloud resources, including 7bridges and Terra. So I think that's all. Just if you want to get started, this is our website. We have a small short paper in cancer research you can read to understand better ideas. Some of those are already outdated, but still. We have free cloud credits that you can apply to experiment with the cloud, with those components of the cloud that are not free, if you're interested. And also we have IDC community office hours. I would be really delighted if you join community office hours. I'm happy to do any demonstrations, answer your questions. I'm there every Tuesday and Wednesday. I spend 1 hour each of those office hours, and I would be very happy to have some company. So please join. I want to acknowledge the technical team of IDC. Without those folks, nothing would happen. And thank you for your attention.
Thanks. Thank you so much, Dr. Fedorov, for the great presentation. And I know that the IDC is really an incredible resource for the broader community. A couple of questions for you. I really enjoyed the framing around the FAIR data principles. I wonder if you could speak to the specific challenges or opportunities related to imaging data that may be unique compared to some of the data that is, components of other CRDC nodes.
Yeah, I admit I don't know enough about challenges in other CRDC nodes to do this comparison. But what we identify is that at the time the data is shared, the submitter may not be aware of what is needed to make the data integrated with other data pieces. The submitter knows only about a subset of use cases for that data set, and they are not motivated to do this harmonization. Sometimes it's as simple as metadata attributes may just be missing, and you do not discover this until you actually try to use the data. In the process of harmonization, we are actually using the data. Sometimes we are the first potential to use this data at all. And the challenge is going through this process, understanding submitter-specific documentation and various files that accompany the data. Very often metadata is separated from the data. Communication with the submitter. Sometimes submitter is no longer around. They may not be reachable anymore at the time that we do the data curation. So I think this speaks for improving the workflows of trying to define what are those metadata attributes that are needed at the time it is submitted, and really coming through with insisting on including this metadata when a submitter is working with the coordinating center. And it's an ongoing process, and I think we're making some progress. There is hope.
Randy, you're muted.
Sorry about that. Next question. Does IDC do any research by analyzing the data, the image data sets?
Yeah, it's interesting you ask about this because yes, within Imaging Data Commons, we have two teams that are funded to actually use IDC for imaging research. We started this by performing replication studies because none of us used the cloud before. And we started by developing a radiomics replication use case, taking existing AI algorithms for segmentation and cancer prediction, replicating them, working with the data in the cloud, doing computation in the cloud. And we do similar processes for digital pathology. I think where we are right now, we are getting close to finalizing the manuscripts that summarize our findings and trying to define best practices, best examples, how to work with the cloud and IDC data. So I really hope that those will be coming really shortly this year.
Great. Next question. Can you speak, and this is combining a couple of questions, but could you speak to the distribution of users on the system today? So academic, industry, wet lab, experimental biologists, computational biologists, and then additionally, if you can comment on the geographical distribution of users as well, that would be.
The first question, no, I cannot comment on this. I do not have that information. IDC does not require any registration. We do not require users to disclose anything about them. We do not track the users. So no, I would be really interested to know the answer, but I don't. About the geographical distribution, we do use Google Analytics. And we have the geographic distribution without tracking individual users. Most of our users are indeed from United States, not surprisingly, I guess. I think Canada and Europe are the next in the list.
If I may, Andrey, what would you ever partner with an institution, a comprehensive cancer center, sort of one-on-one? Say we wanted to work with you and upload sort of all our images for.
No.
Yeah.
Well, you know, if individual center, I can't imagine a scenario where a center would want to upload all of their images and make them public. That's probably never going to happen. But remember that all of the data we curate must be public.
Well, we do that for some radiological images. We do have partnerships where we upload them all in partnership. So that's.
It would be very interesting to discuss, I think.
Okay.
If they indeed want to release it publicly, we should initiate communication and involve our NCI stakeholders.
Just a brainstorming kind of thought. Thanks.
Yeah.
Maybe just following up on that conversation, can you speak to any data access controls on the data?
Yes, I can. All of the data is publicly available. There is no access control at all right now. That's the current situation. The data is available in Google Cloud buckets under all authorized users access. So you need to have a Google ID to access the data, but your Google ID is not tracked. No one is tracking you. You don't need to provide any bill information. Egress is free, no strings attached. Going forward, this may change. Going forward, we are discussing possibility of supporting limited access and controlled access data. Timing of that and decision on that is pending.
Okay, maybe last question here. Do you have a feeling for how much of the usage is for experimenting, training deep learning models versus other uses?
Again, I wish I knew, but I just don't have those insights into our user community, unfortunately.
Okay, so with that, I think that wraps up our Q&A session. Thank you, Dr. Fedorov, for a great
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4-CANCER.